Hey everyone, here we go for our next lecture, and this one is on more anatomy of the peripheral auditory system, but this time on the inner ear, which is the structure we call the cochlea, which is derives from, I think, the Greek word for a snail shell, so similar to the concha in the pinna, the cochlea. So we're going to look at the structures that make that up, kind of understand its shape and some of those structures, and just get a basic idea of describing what its response to sound is. And then later, as I said in the last video, we're going to come back once we have built up some tools, kind of mathematical concepts and some simple physics to understand why it behaves that way. So here we're just going to kind of describe its behavior, and later we'll try to understand, like, how does it pull that off? What's the basis of how it makes that happen? Okay, so I'll show you here an image, and there's a description later on of where I got this from in the PowerPoint slides. So this is actually a dissection of an actual human cochlea from a human temporal bone. And what you can see is the people who prepared this have kind of chipped away at the temporal bone um, until they've kind of broken through into the inner cavity of the cochlea, and you can see that it's a spiral. And we'll talk more about the shape and the structures here, but the main point I want to make, and they mention this in the textbook quite a bit, so if you like check in chapter 16 and a couple other places, they point out that often we draw the cochlea as if it is a freestanding thing, like a snail shell, right? But really it's not. Like, you can't take the cochlea out of, it's not like a kidney, right? You could remove a kidney from a body. You can't really remove the cochlea as a thing, because it's just a hole. It's a cavity in the temporal bone that has a special shape, and it's filled with these tissues and cells. Right, but it's not a freestanding thing as it's often drawn, and it's kind of tricky to draw that. But I think this gives the idea that that it's sort of embedded in this bone. The bone is its walls, and we can sort of see what the inside is like. Okay, so if we go back to our drawing of the three divisions of the peripheral auditory system, we've already talked about the outer ear, and the middle ear, and now we're going to be talking about the inner ear, the cochlea. Okay, so just as I just said, that we often draw it as if it's a freestanding thing, even though it's not. But imagine that we could kind of visualize the surface of it, and one way to think of it is if you took some flexible tubing and coiled it up. Oh, wait, I have some flexible tubing right here. Right. Um, okay, so if you kind of coiled this up like this, and then you stuck it in a bucket and you filled it up with plaster so that it was just like a solid thing, then the plaster would be like your temporal bone, and the inside of this coil would be the cochlea. Obviously, it's not exactly the shape. But if we could sort of chop across that, as is shown in this picture, then we can see, you could sort of see that you would chop through this single tube that's been coiled up several times. Okay, so that's what this is showing. And so now we can see that each of these turns of the cochlea is going to get dissected if we did this. And we can see that inside that main tube, there's three divisions. And we'll see what the names of those are in a sec. And these are divided by membranes that stretch across from the inside to the outside of the spiral as it goes up. And sitting in here are the cells of the inner ear that are going to convert a mechanical vibration to an electrical signal and then to a nerve impulse. And these kind of yellowish brown shaded things here are showing where the nerve fibers and the neurons and the cochlear nerve, which is kind of the bundle of all of those things that comes out of the cochlea, are. So that's where those locations are. So we'll look at that in more detail as we go ahead. So here, kind of zooming in on this section here, inside the cochlea, um, we see these three divisions again. So the one, well, where shall we start? Okay, so we've got the basilar membrane here, the base membrane. So that divides the whole duct in one place, and then there's another membrane up here called Reissner's membrane, and that divides it again, and that makes these three compartments. Okay? And each of those is called a scala, which just means comes from a like staircase, like a spiral staircase. And the one at the bottom, under the basilar membrane, is called scala tympani. The one at the top is called scala vestibuli, and the one in between is called scala media, which just means the one in the middle. Okay, and this is where the good stuff goes on. So inside scala media is a complicated structure called the organ of Corti, just named after the anatomist who first described it. And that's sitting on top of the basilar membrane, and it's here that we have the various cells. We'll see they're called hair cells that react to vibration and convert that vibration to a electrical signal and then a neural signal. And then here is, it shouldn't really be called the auditory nerve because the nerve is the bundle of all of these nerve fibers, but this is like one nerve fiber that's heading out from here into the middle of the cochlea. And we'll see if there's names for all of these parts. Okay. The other thing I want to point out in this diagram is the stria vascularis. So that's vascular, it's got blood vessels. So this is the kind of metabolic supply of nutrients and oxygen comes in through the blood vessels that flow through this tissue here. Okay. Okay, here we go. So imagine that we could kind of unroll this, and we'll see in a lot of our pictures where we're trying to understand the function of the cochlea that we won't worry too much about that it's coiled up into a spiral. And we'll imagine that we can just kind of flatten it out, okay? So here it's kind of drawn partly uncoiled. And again, we can see these three compartments. So there's scala vestibuli, which is actually connected to the vestibular system, which is for balance and perception of acceleration, right? And the stapes, which we saw in the last set of slides, is sitting on top of the oval window. So when the stapes pushes in and out, it's pushing on the first thing it's doing is pushing on this fluid in scala vestibuli. So that's the fluid that's above scala media and the basilar membrane. Below, so the scala media is the shaded in thing, and that's a sealed off compartment. So at the top, there's Reissner's membrane. At the bottom, there's the basilar membrane. And as you go up the spiral, up and up and up, spiral, when you go up to the very top, those two things kind of connect to each other and they seal it off. So this thing is its own little tube, right? It's not connected to the other ones. And in the bone, there's a hole here that connects those two cavities, scala media, or scala vestibuli and scala tympani are connected together like right up at the top of the spiral. And that connection where the fluid spaces connect is called the helicotrema. We can see down here so scala vestibuli is where at the base, down at the bottom, nearest the middle ear, that's connected, or it's terminated by the oval window, right? And then scala tympani is terminated by the round window, which we saw in the last set of slides. You can actually, if you could get into the middle ear cavity, you could actually see that membrane covering the round window there. And as we said, that's the kind of the pressure relief. So when the stapes pushes in, that kind of bulges out and allows us to get energy into that system. Okay. So what I'm going to show you a bunch of pictures, and then we'll talk about the function of these things and how the cochlea responds to sounds. Okay. So what we have here is a series of CT scan, so it's like 3D X-rays, and these are slices like horizontally across the head. So in this one, you can see the nose and the eyeballs and the skull. Um, and what these are a series of slices that are kind of getting higher and higher. And what we're going to do is we're going to scan through the middle ear cavity, and we'll actually be able to see that cavity where the ossicles are, and also some of the turns of the cochlea, right? I don't know what the resolution is going to be like on the video, but if you get the slides, then you can look at these sort of in higher res and see what's happening if it doesn't work out on the video itself. Okay, so this is the lowest image, and we can just, these dark areas are just holes in the skull. So that's the cavity, the middle ear cavity. And here we can see kind of this kind of spongy, there's a lot of air cavities in the skull in this area here. And then there's kind of more solid bone here around the back. So the next slice is going to be a little bit higher up. And what you'll see is in this one, you can make out the bottom turn of the cochlea is this dark area here. And you can see the same thing on the other side. And so this gives you an idea of how big it is, like say compared to the eyeball, and how, that it's quite far inside from the edge of your skull. Here we can see the soft tissues of the pinna. And here is the ear canal, the external auditory meatus. Not sure if that came out better in the last slide. Yeah, you can kind of see that's the ear canal coming in there. All right, so we're going to go up again, and now we can see a couple of turns. It's not great resolution, right? But you can kind of see that you're seeing multiple of these turns like we saw in that drawing of the blue freestanding cochlea sliced in half. Okay, so we can see the cochlea, and that again gives you an idea of how big it is. And here you can actually see the malleus and the incus in this image. This is kind of zoomed in here, so those are showing up. And then here we can see the internal auditory meatus. So that's the tunnel that lets the nerve fibers, the auditory nerve, vestibular nerve, and the facial nerve, at least, go through this into the brain stem, right? So the bottom part of your brain and your cerebellum are sitting down here in this area. Okay, we'll go up one more time. And here we can see actually one of the semicircular canals of the vestibular system. And now we can really clearly see the internal auditory meatus. And we can see the malleus and incus again in the middle ear. Okay, so that kind of gives you an idea of how well protected the structure is. Like it's well inside the lateral edge of your skull, and then also how big it is. So again, looking at this preparation, and if you want to, I downloaded these a while ago, so I don't know if it's still available at this address, but this is where I got it from. So in this preparation, they've taken a temporal bone dissected from a deceased human donor and just chipped away very finely. So you can see here that we're starting, like the bone that's left here is very, very thin. And here they've actually broken through and they've added some staining so that we can see different structures inside the cochlea. This is kind of looking down on top of the spiral structure. And the light colored area here is the basilar membrane. You can see that's kind of spiraling up. And then these dark colored things are actually the nerve fibers that are running out from the organ of Corti to the interior, the middle part of the cochlea. Okay, if we zoom in right at the top, we can see that we kind of come to the top of the spiral staircase. And this lets us see a special structure here. So some of the soft things have been taken away in this picture on the left. And what we see here labeled as the OSL is the osseous spiral lamina. Okay, so it's the bony spiral flat thing. It's like a spiral staircase. And that forms the inside part, like the inner part of the turns of the cochlea. So if I just go back to the previous slide, all of this, so that down at the bottom here, this is where the brown things are. These nerve fibers are running through this bony shelf that kind of sticks out. And then the organ of Corti is sitting on top of the basilar membrane that's kind of stretched across this space. So that's not bony. That's something that is soft and can vibrate up and down. And so this shelf spirals up on the inside part all the way up to the top. And we call that the apex of the cochlea when you get right up to the top. And that's where the helicotrema is. If we had all the tissues there, then that's where Reissner's membrane and the basilar membrane would meet and kind of pinch off scala media. So it's completely separate from the other bits. Okay, so that's right up at the top. And then down near the base, the bottom of the cochlea, so nearest the stapes in the middle ear, we can see again these nerve fibers running through the osseous spiral lamina, so the bony spiral flat thing. And then this is OC is the organ of Corti, so that's sitting on top of the basilar membrane. So that's kind of stretched between the edge of the osseous spiral lamina and the outer part of the spiral tube, right? If we took like kind of a side view of the very apex of the cochlea, again, there's been some stuff removed here, right? So let's see if I can draw this. They've taken away the bone, that would be here, right? And so the basilar membrane would be across like that, and Reissner's membrane would be kind of like this, and the organ of Corti would be sitting there, and here's the nerve fibers, right? So we get a really good view of the kind of the spiral shape of the osseospiral lamina, and these brown things again are stained nerve fibers, and what happens to them is they're heading in this direction, and then they all kind of dive in, whoops, to the middle. So from over here, like that, and up here they dive in, and down there they dive in. So you kind of get this spiral rope of nerve fibers that run down the center core of the cochlea that's hollow, and that's called the modilus. Okay, so that central part of the cochlea carries the nerve fibers that altogether make up the nerve itself out to the rest of the brain. So I'm just going to show you a few more pictures. So this is a kind of chipped away cross-section of just the bony parts. This isn't a human cochlea, and I can tell that because it's quite tall, whereas human ones are more sort of flat. But here you can see quite clearly down at the bottom where the stapes is on the round window and how that would be connected to scala vestibuli that's on top of the scala media and the osseous spiral lamina, and here's the round window down at the bottom connected to scala tympani. Right? So you could imagine if you guys are familiar with the Magic School Bus books, if you could get in a tiny submarine and go in here, then you'd be able to kind of drive your submarine all the way up scala vestibuli, all the way up to the top, and then we get up to the helicotrema up here, and then you'd be able to come back down underneath the osseous spiral lamina in scala tympani until you got back down to the round window here. Okay, and then down the core of this is hollow, and that's where the nerve fibers run out, so they'd come out down this way. All right, so just taking a closer look at some of these membranes in an actual microscope slide. We can see again the basilar membrane here. Okay, organ of Corti sitting on top of that. Stria vascularis, it's got the blood vessels. The spiral ligament is this thing that kind of sticks out that the other edge of the basilar membrane is attached to. Scala tympani is down here. The fluid that's in there is called the perilymph. We won't worry about the fluids yet. We're going to talk about that when we get back to kind of the electrical setup of the cochlea in a while. Scala vestibuli is up here, and then scala media in between. And the fluid in there is called endolymph. I'm not sure these are spelled right. Maybe that is right. Let's check that out. And that's a different, the concentration of stuff in there is, it has different ions dissolved in it, and we'll see why that's important later on. Okay. What's labeled? Oh, the thing that's labeled on here that we haven't seen before is the spiral ganglion. Okay, so these are all the cell bodies of the neurons whose axons or nerve fibers are connected to the hair cells in the organ of Corti, right? So this is kind of like yet another tube that travels up in parallel to the main tube, but on the inside. And that's where all these neurons live. And they send their axons out into the medullus, right, into the center here, and they're just going to shoot out this way. I was just talking about the helicotrema. All right. So we're looking again at the organ of Corti. So let's kind of zoom in on that and see what some of the structures in there might be. Okay, so again, in this picture, the neural stuff is yellow, but now we're going to kind of zoom in on what the organ of Corti is made of. So here's a drawing that shows us some of the parts. So this, if we could kind of, again, chop through one turn of the cochlea, we'd see these ducts, scala vestibuli, scala media in the middle, and scala tympani. Here we see the nerve fibers coming out, and this is the organ of Corti here that's built up some special cells and this interesting membrane that kind of sits on top of them. All right, so if we kind of zoom in here, we can see that what's sitting on top of the basilar membrane, so that's this thing down at the bottom, are a bunch of different cell types that, and some of them we're not going to worry about. Some of those just make up the structure. So if on some of the slides it's labeling Henson cells and Dieter cells, don't worry about that. But we're just going to talk about the hair cells. So there's our cells whose bodies are embedded in the organ of Corti, and they have these little bristles that stick out at the top called stereocilia, and those are really important for converting the mechanical motion of the basilar membrane. So in response to sound, this is actually going to vibrate up and down. And the motion here is a bit complicated, but we'll take a look at that in a sec. Sitting on top, or kind of laid over the top of the hair cells and their bristles, or stereocilia, is this kind of jelly-like membrane called the tectorial membrane. And there's two kinds of hair cells. There are 3 rows of outer hair cells over here, and there's 1 row of inner hair cells. And we'll see later that the main role of the outer hair cells is they actually work like little motors, or they actually can change their shape in response to sounds. And that can enhance the vibration of the basilar membrane. The inner hair cells don't do that, but they're kind of along for the ride, but these are the ones that pick up the vibration, and 95% of the nerve fibers that come into the brain in the auditory nerve are connected to these inner hair cells. So these ones are their main job is to convert the vibration into electrical signal and then a neural signal. And these ones have some similarities in the way that they work, but their job is to enhance the vibration actively. Okay. Here's just another drawing showing that. So here we have the inner hair cells over here, and the 3 rows of outer hair cells over here. So this on this side would be the towards the medullus of the cochlea, right, towards the inside part of the spiral, and this would be towards the outside part of the spiral. So here we can see the Dieter cells and Henson cells labeled. Don't worry about that. We're just going to worry about where the hair cells are. So there's 3 rows of outers, and that means there's more of them. So in one cochlea, there might be like 12,000 outers and maybe only 35 inners. Okay. If we just look at some drawings here, we can see that these guys have different shapes. So the inner hair cell has a more kind of eggplanty, bulbous sort of shape, and it has more nerve fibers connected to it. So we can see these are showing where the nerve fibers are picking up. What they're going to pick up is neurotransmitters that are released by the inner hair cells in response to sound. And here we see an outer hair cell, which is more kind of straight up and down cylinder. And it also has nerve fibers connected to it, but it has this different shape. And we'll see later that there's special proteins in that cell that make make it actually be able to mechanically vibrate itself. And on top of each one, the only parts that's sticking out into the fluid in scala media, that's the endolymph, are these stereocilia that are in, there's some close-ups, but you can see they have a special arrangement. All right, so here's an actual slide. It's a little hard to make out what we're looking at, and it's a bit fuzzy, but if we kind of follow this outline here, draw, this is an outline of an inner hair cell that's kind of leaning over. Sorry about those zaps. And here's the kind of top plate of the inner hair cell, and here's its stereocilia sticking up into the endolymph in scala media. Okay, and then over here is one. Oh, I need to get a better mouse, don't I? That's one outer hair cell, and you can just see a bit of its stereocilia sticking up here also. Okay, and in this picture, the tectorial membrane's being removed, but that would be sort of sitting over top of everything like that. So if you zoom in right on the stereocilia, we can see that they are just not a random bunch of bristles, but they have an arrangement, and they're usually arranged in 2 or 3 rows. On the outer hair cells tend to have 3 rows, kind of in this V shape. So there's kind of tall ones, and shorter ones, and even shorter ones. And not quite the same arrangement on the inner hair cells, but they do have sort of taller ones and shorter ones, and would have 2 or 3 rows on those ones as well. And what happens is that these rows of stereocilia are actually connected together. It's a little hard to see, but there's these tiny protein filaments that connect them together. Those are called tip links. That's tip links here. Okay, so they're actually connected together by these stretchy proteins, and we'll see later on that that's important for their function in converting vibrations into electrical signals. Well, so just to quickly tell that story, if we imagine that the organ of Corti is going up and down, and the tectorial membrane is kind of sitting on top of the stereocilia, when things go up, the stereocilia are going to get pushed in one direction, and when it goes down, they're going to get pushed in the opposite direction. And when they're pushed over in the direction of the tall ones, these tip links are going to be put under tension, and it's actually going to open some complicated protein ion channels on the walls of these bristles, right, that are actually hollow protein tubes. And that allows ions to flow into the inside of the cell and change the voltage. So we're going to go through all that later on. So I'll just show you a few more pictures here. So this is looking down on top of the organ of Corti here, and you can see the 1 row of inner hair cells. But all you can see of the hair cells here is just the stereocilia, these white things that are sticking up, and the bodies of those cells are down in the main part of the organ of Corti. And here we can see the 3 rows of outer hair cells, and here we can see the wall, the outer wall of the cochlear spiral, right? Here's the what's being done in this picture is they've actually cut off the tectorial membrane. So here's the edge where they've taken away the tectorial membrane, because there should be the tectorial membrane kind of lying over this whole structure, right, like that. And it's important because that's the thing, that's the stereocilia from the outer hair cells are embedded in the tectorial membrane. So when everything goes up and down, there's this kind of shearing action that bends them back and forth. And for the inner hair cells, the stereocilia are just kind of sitting, for the most part, in the fluid, and they're kind of getting wafted back and forth, kind of like seaweed in a wave, right? But that's what bends them back and forth, is the fluid motion there. All right, if we just look closely again, we can see the 3 rows. Now we're looking like straight down on top of the organ of Corti without the tectorial membrane. And we can see these 3 rows of outer hair cells. we can see the whole top of the outer hair cell here. So I'll just kind of outline one of these guys. That was terrible, but you get the idea. And you can see these kind of V-shaped arrangements of the stereocilia. So there'd be 3 rows of those, with the tallest ones kind of pointing towards the outside. And over here we have the inner hair cell. So again, here's like the top of one inner hair cell, and here's the tall stereocilia, and there'd be at least 1 row of shorter ones towards the inside part of the spiral there. All right, so this might be the last picture of this series, but here they've kind of chopped through the organ of Corti itself, and so now we can see the top of the cells. So let me outline one of those. So here's the top of an outer hair cell and its stereocilia, but now we can also see its cell body there, and we can see the body of its friends in the middle row and the inner row, right? So they would have their tops here like that, and this kind of repeats all the way down to the base and up to the apex of the cochlea. And here we see the inner hair cell kind of, it's a little harder to see, but again, it's kind of leaning over, and then its top is there. Well, that was bad sketching, but you can see where the stereocilia of that one are. And again, the tectorial membrane's being snipped off here, right? This would continue over and be lying on top of the rest of that. Okay, so let's, the last bit we're going to do here is just kind of wrap up with looking at how the cochlea responds to sounds. And later, and I don't want to say how, I want to say that we're describing what the response is like. And later, we'll talk about how it does that, like why is it like that, okay? And just to get started, let's look at this little computer animation. And here's the YouTube link if you want to see the original. So what they're doing in this animation is you'll see that we kind of fly into the ear, and then we're going to imagine that we unroll the cochlea into a straight line and then kind of disappear it and only look at what the basilar membrane is doing. So we'll just imagine that nothing else is there. Obviously all that stuff needs to be there to make it work properly. But we're just going to look at what the basilar membrane is doing and not worry about how it got to do that. So let's play this, and I might stop and interrupt and say a few things as we go. Okay, so we're gonna fly down the ear canal. There's the eardrum. We're magically going through. Okay, so we know we can't really see the cochlea like this, but we're imagining that it's a freestanding thing. We unroll it and then disappear everything except the basilar membrane. And here comes some sound. Okay, so I'm just gonna pause that. Maybe I'll just go back a few seconds. So I want you to remember that this end up here is nearest the base. So that's near where the stapes is, okay? And this end down here is at the apex, okay? And you can see that there's some details about the basilar membrane's dimensions that are different, right? That it's narrow near the base and wide near the apex, near the top. And we'll see later that's important for kind of tuning its frequency response. But what I want you to pay attention to is which part of the basilar membrane vibrates when we have these 3 different tones, these 3 different pitches that are played. I'm just going to wind back a little bit, and we'll go from there. Okay, so you might have noticed that when they played the low pitch by itself, there was a little patch of the basilar membrane that vibrated a lot here. And then when they played the medium one, it was sort of in the middle, and when they played the higher pitched one, it was down towards the base. Okay, so there's this tuning of the frequency sensitivity along the basilar membrane, which we'll see is quite important um, uh, for coding sound and representing sound all the way, throughout the auditory system, from the periphery all the way up to auditory cortex. Um, and you also see that when they play multiple sounds at once, then lots of different parts of the basilar membrane vibrate. So obviously, those first three sounds were just pure tones, one single frequency. But you very rarely encounter a sound like that in the real world. Instead, we have complicated sounds like a musical instrument or speech that has tons of different frequencies in it. And when there's lots of different frequencies playing, then lots of different parts of the basilar membrane are going to vibrate. So we'll just let this go. A bunch of low notes vibrate the apex. High notes come in. More of the bass vibrates. And then when the sound stops, the vibrations die away. So that was kind of cool. Here's a, a less cool animation of what's happening there. And again, we're going to kind of explain later why it behaves like this, but what's going on. So we've got a sound coming in to the ear canal. It's going to push on the eardrum, the tympanic membrane. It's going to make the ossicles vibrate. The stapes is going to push on the fluid here, and that's going to create a kind of an oscillating pressure difference between across the basilar membrane. So there's going to be a different pressure in scala vestibuli than in scala tympani, right? And if we have high pressure up here and low pressure down there, that's going to push the basilar membrane down. And if it's the other way, it's going to get pushed up. And it turns out that so here you can see a cross section showing the basilar membrane going up and down, right? The purple shading is scala media. And if we kind of look along the length, if we just draw the basilar membrane again from the side, we can see that we get what's called the traveling wave, and we'll talk more about its characteristics later. But it's sort of restricted to, there's only one portion of the basilar membrane for this one particular frequency that's being played in that gets a lot of vibration happening, right? And if we change to a lower frequency, which part of the basilar membrane would vibrate? Well, if you remember from the video, that lower frequencies would make the vibration happen up here. And if we change it to a very high frequency, a high pitch, then we would get more of this vibration. Still the same similar shape, but wiggling faster, but it would take up about the same amount of space. Here at the base for high frequencies, and up at the apex for really low frequencies. So this is kind of nifty. This is the way that the auditory system spreads the frequencies out, that divides the frequencies one from the other, so they can be analyzed by separate parts of the organ of Corti and get separately into the auditory system. So you can think of a little bit like an acoustic prism, right? That this setup divides a sound that's made up of lots of different frequencies and puts them in different places. Okay, and that principle is called tonotopy, which I don't think I have written on here. So tono, T-O-N-O, topy, T-O-P-Y. So topy, like in topographic map, it's just, so it's putting tones in different places. And we'll see that that tonotopic principle applies to lots of places in the auditory system, as I mentioned earlier. Okay, so here's just quickly, and we'll see these figures again when we really dig into the mechanics of this, but we can see that if we look at just kind of in this computer simulation, just imagining what the basilar membrane is doing, if we play a high frequency tone, so this is a tone that's vibrating at 4,000 times a second, okay, or here's one that's vibrating at more slowly, 1,000 times a second, then this patch of vibration would be more towards the apex, and then a lower frequency tone, 250, would be even further up towards the apex. So different frequencies make different parts of the basilar membrane vibrate, and that's important for the way sound is processed and the way that we perceive sounds. I just should say that these these diagrams and this one here, if we go back, right, are vastly exaggerating how much the basal membrane moves up and down, right? This shows like that the basal membrane is almost touching the wall of the cochlea. And it's drawn like that because if we drew it to scale, you wouldn't be able to see anything because the amount of the vibration that it goes up and down is very, very tiny compared to the dimensions of the cochlea, right? So don't think that if you play a loud sound that the basal membrane is going to be like hitting the sides of the cochlea. It doesn't move that much at all, but it's being exaggerated here so we can see what's going on. All right. So just to kind of lay out this principle, again, you will see this figure again, that we have a mapping of different frequencies onto different parts of the basilar membrane. So imagine we could go into the cochlea and with a tiny pair of scissors and just snip out the basilar membrane itself and just lay it down flat on a piece of paper or something. We would see a couple of things. One would be that it's kind of narrow near the base and wider near the apex, which was also shown in that movie animation. And we would see that if we play different tones in and saw where it vibrated the most for each of those tones, we could kind of get a map and we could label each piece of the basilar membrane with that frequency. And we would see that high frequencies, or the the basal part would get labeled with high frequencies, and the apical part would get labeled with low frequencies. The other thing we would see is that the frequencies change more rapidly near the base than they do at the apex. So like from here to there, from this spot to that spot, is a 13,000 hertz difference, right? But about the same distance from here to here, up near the apex, is only a 1,500 hertz, 1,500 cycles per second difference. And so we'll see later that this means that the map of frequency is not a linear map, that high frequencies are kind of scrunched together and low frequencies are more spread out. And that's the way the frequency tuning goes. Okay, so that's on the very last slide, I just want to, there's two more. So let's go back to this drawing. And what I want to show you is kind of how this looks in real life. So let's imagine that we're looking at the cochlea here, and instead of just seeing the basilar membrane, let's imagine that what we can see is just the hair cells and the nerve fibers that make up the stuff that's being colored yellow here. Okay, so we're going to be able to see the hair cells that actually are missing from this drawing, and these nerve fibers. So this is a nice image, and I just realized I've forgotten to put the lab name that I got this from on this slide. So I'll fix that up before I post the slides themselves. But so this is actually from a mouse cochlea, and imagine that you're kind of looking from the base, kind of up the modulus towards the apex. Okay, so we got underneath the cochlea and we're looking up, and in this image, they have stained the hair cells green, and you can see the 3 rows of outer hair cells here, and the 1 row of inner hair cells there, somewhat separated from them. And they've stained the like the axons, the nerve fibers, and the neuron cell bodies red. Okay, so what you can see is these are the nerve fibers here that in those original dissection drawings were stained brown, right? So you can see them coming in. And this kind of lumpy rope here, these are all the cell bodies of those neurons that are connected to by these axons, right? So these guys are all living, you can't see the bone in this image, but these are all living in that secondary bony tunnel. That's the spiral ganglion, and that is going, so we're kind of looking from the bottom to the top, right? And we're seeing the spiral of hair cells kind of spiraling up away from us and getting tighter and tighter. And we see the spiral ganglion where those cell bodies live, spiraling up and getting away from us up towards the top. But coming back towards us, like coming out of the screen, are the nerve fibers that are sort of the outputs of these neurons, right, that are forming this bundle of nerve fibers all together like a rope, and that's the auditory nerve that's coming out of the cochlea and going through the internal auditory meatus and on to the brainstem. Okay, so that's finished our tour of the anatomy of the inner ear. So I mentioned there might be some changes to the syllabus, and our first change is that I think that's enough anatomy, so I'm not going to worry about the central anatomy right now, and we'll just leave that towards the end of the course where we can kind of link it together with some of the functional stuff that we've talked about along the way. So I will leave this there, and I will see you again in my next video, which is going to be about what is sound and how can we describe simple uh, vibrations that make sounds. All right, see you soon.